ucapkan di seminar Rabuan kerjasama dengan Departemen Biostatistik, Epidemiologi dan Kesehatan Populasi Fakultas Kedokteran, Kesehatan Masyarakat dan Keperawatan Universitas Gajah Mada. Perkenalkan, nama saya Karisma Dewi. Pada kesempatan kali ini saya akan dengan senang hati memandu jalannya acara seminar Rabuan. Sebelum kita melanjutkan acara, tanpa mengurangi rasa hormat demi kelancaran acara pada kali ini, kami ingatkan kepada Bapak Ibu untuk bisa menonaktifkan mikrofon selama acara berlangsung, kecuali nanti pada saat sesi tanya jawab dan sudah diizinkan oleh moderator. Bapak Ibu yang kami hormati, seminar Rabuan kali ini mengangkat tema yang luar biasa menarik, SARS-CoV-2 Surveillance Using Wastewater and Environmental Sampling in Indonesia, dengan pembicara-pembicara yang luar biasa pastinya. Dr. Tiara Martias, Master of Public Health, Doctor of Philosophy, akan memimpin diskusi pada kesempatan kali ini. Sedikit informasi mengenai beliau. Mendapatkan gelar dokter di tahun 2008 dari Universitas Gajah Mada, kemudian di tahun 2011 beliau mendapatkan gelar Master of Public Health dari the University of Melbourne, dan baru saja kembali dari University of Melbourne juga mendapatkan gelar Doctor of Philosophy dari the Nossal Institute for Global Health. Tanpa menunggu lebih lama lagi, Bapak Ibu, mari kita sambut Dr. Tiara Martias, Master of Public Health, Doctor of Philosophy. Monggo, Dokter.
Thank you, Mbak Karisma Dewi. Selamat pagi semuanya, Bapak Ibu, apa kabar? Semoga oke-oke. Ini disclaimer sedikit karena pembicaranya juga ada yang dari luar negeri dan bahan-bahannya juga dari luar negeri. Kita bahasa Inggris ya hari ini ya. Jadi ya nanti kalau ada pertanyaan pas Q&A session, itu bisa diketikkan atau langsung ditanyakan dalam bahasa, terserah bahasa apa asal bukan bahasa Jawa, karena saya nggak bisa bahasa Jawa. Oke, terima kasih banyak Mbak Karisma. Oke, terima kasih juga untuk perkenalannya. So good morning everyone. We have here all three presenters that will be sharing their knowledge and point of discussion that will be interesting for today. As Karisma Dewi have said, I'm Tiara Martias. I'm currently with the Health Policy and Management Department. And welcome everyone. And also I would like to welcome Professor Julie Bynes, Dr. Indah Kartika, and also Dr. Fika. And thank you so much for joining us today. And without further ado, I would like to introduce our first speaker, Professor Julie Bynes. Hi, Julie. How are you?
Good. All good. Thank you very much. And thank you for that kind welcome. It's lovely to be here.
Cool. Thank you so much. Yes, so she has a very long-standing experiences in pediatric gastroenterology, so I won't try to read all 35 of your CV, but I just would like to highlight that Professor Julie Bynes is currently a pediatric gastroenterologist at the Royal Children's Hospital in Melbourne, where she leads the clinical nutrition program. She's also the deputy at the center, and the most interesting one that we might all have heard about is that she is the one who's leading the rotavirus vaccine program, which is also currently happening in Indonesia with our pediatric program at the Sarjito Hospital. And Professor Julie Bynes is a really, really big name, and I'm sure all of you would have known by now. And she will be presenting to us on why we can use wastewater as one of the surveillance technique for COVID-19 from the clinical point of view and gastroenterology point of view. And then Dr. Indah Kartika will then explain what's happening in Indonesia now with wastewater surveillance, and Dr. Fika would then explain the way forward. So we really look forward to your presentation, Professor Bynes, and the Zoom is all yours.
Thank you very much for that kind welcome, and I'll share the screen now. Hopefully you'll be able to see my presentation in a moment. I'll just bring that
all
up.
good, Prof.
Yep, I'll just bring it to, is that good?
Yes, perfect.
Excellent, thank you very much. Well, my role today is to set the scene on why you might like to consider wastewater surveillance to support the public health response during the COVID pandemic, and then introduce the your two terrific speakers to follow me to have a look at some of the possibilities of this relatively new modality. So you're very aware by now that SARS-CoV-2 invades a lot of the organ systems and influences a lot of the organ systems of the body. And the gastrointestinal tract is no different to that. Certainly, and we'll just talk about the impact of SARS-CoV-2 infection and inflammation on the gut. And considering that the main modality of infection involves the ACE2 receptor, and the expression of the ACE2 receptor is across many of the organ systems of the body, it's not surprising we do see the impact of SARS-CoV-2 infection throughout the body. Gastrointestinal symptoms are very common in COVID disease, and in adults it's thought that about 17% or about that of patients presenting with COVID have some symptoms of gastrointestinal tract. The most common is an anorexia, but also nausea, diarrhea, abdominal pain are common in patients with COVID disease. Sorry about that. I'll just go forward again. Some reports are up to 50% of infected patients that have GI symptoms at some stage during the course of their illness. In children, this seems to be a little higher, and diarrhea is often a presenting symptom of COVID disease. Severe disease appears to be more common in patients that do exhibit gastrointestinal symptoms. But on the other hand, up to 28% of those with gastrointestinal symptoms and a positive RNA in the stool do not have respiratory disease, respiratory symptoms. So you can have gastrointestinal symptoms alone and COVID positivity without respiratory symptoms. And that's important when we're looking at the way of managing and presenting with GI symptoms. You'd still need to think of SARS-CoV-2 infection in that cohort. Shedding of viral RNA in the faeces is common, occurs early in the illness and can be prolonged. And the incidence of finding RNA in the faeces can be detected within 3 days of infection, any time, up to 2 weeks. It can be present before the onset of respiratory illness and after the nasopharyngeal swabs are negative. About 15% of participants, these are adults who present with a positive nasopharyngeal swab, have viral RNA in the stool on admission, including those with no gastrointestinal symptoms. But about 48% of those during the course of their COVID illness will test positive in the stool. And only about 50% of those patients with the presence of RNA in the stool had GI symptoms. The duration of shedding is quite variable. About 70% of positive stool RNA is present despite negative respiratory samples and can exist for up to and beyond 33 days following the diagnosis of COVID. And the mean duration of positivity in the stools is about 28 days or so. And disease severity is not associated with prolonged shedding in the stool. In this recent or relatively recent review, this looks at the difference between adult patients and paediatric patients. And I suppose I'd just like to, as a paediatrician, just point out that rectal swab positivity or stool positivity in children seems to be more common than adult at the time of diagnosis. And the duration of their positivity appears to be longer than in adults. And in small numbers of studies, but the presence of a positive stool after the nasopharyngeal swab is negative is very common in children compared to adults. So it raises the question, is there something different between children and adults? This is a timeline you may have seen before, which looks at this main line in the middle here is the symptom onset, and when various different tests test positive with respect to the virological tests, the nasopharyngeal swab are usually taken at the time of symptom onset, tends to continue as positivity, but for the first couple of weeks and then drops. This is the same for bronchoalveolar lavage, which drops very quickly in the first week or two after onset of symptoms. On the other hand, the stool PCR, which is shown in the yellow, appears to occur a few days before symptom onsets. That's respiratory symptom onset usually, peaks around 12 days or so, and then continues much more prolonged, even after the onset of when you start seeing antibody positivity and nasopharyngeal swab negativity. The mechanism for gut infection with SARS-CoV-2, as well as SARS-CoV, the original SARS, is via the ACE receptor, and the SARS-CoV-2 tends to bind more efficiently to this ACE receptor than the original SARS virus. It suggests various immunofluorescence screening suggests that ACE2 receptors are abundantly expressed in glandular tissue of the gastric, duodenal, and rectal epithelium, and you can see the virus appearing in staining of the epithelium of the intestinal tract. And when you put SARS-CoV-2 virus in intestinal organoids, you see active infection of those organoids. And the mechanism of infection is thought to include a variety of different elements in the epithelial border. Firstly, entry, replication, and shedding in the gut, developing an immune response within the intestinal lining, promoting an immune cell inflammation in the various different other elements of the lymphatics and circulating blood volume, and also altering the microbiome composition. But what are the implications for transmission of infection? If we know there's a lot of gut symptoms, there is replication in the gut. Does that mean that the stool is infectious? And that's been a bit more controversial. The detection of viral RNA does not necessarily mean that there are viable infectious virions present, or that the virus can be spread through fecal transmission. However, it has been seen in a small number of cases and also in the laboratory, particularly with organelle cultures. The other question is whether fecal oral or aerosol transmission may contribute anyway to close contact transmission in households, nursing homes, childcare, schools, or nosocomial acquisition in healthcare. And how important is this in areas where informal sanitation systems exist and there is risk of environmental contamination? And hence the idea of looking at sewage as a marker of surveillance for SARS-CoV-2, particularly in areas where it's not feasible to do individual swab testing of the community. And this first was identified or pursued in the Netherlands, where they found that there was SARS-CoV-2 present in the wastewater from the major airport 4 days after the confirmation of the country's first case of COVID-19. And when they then monitored wastewater systems throughout the regions in the Netherlands, they could accurately identify the regions as the cases occurred. And so it raised the possibility of an early warning system. This wastewater surveillance is now being applied in various areas of the US, Europe, Japan, and Australia. And the reported detection in wastewater and formal sewage systems is about 1 case per 100,000 population. However, the longevity of the SARS-CoV-2 in the wastewater is less clearly known. Thankfully, bleach and chlorine treatment appears to be effective in getting rid of any active virus. But the question is, can we use this as a mechanism for surveillance? And I just took an example of this and as an introduction to what Inda and Vika might be talking about in just a moment, is in Victoria, this state I belong, each day they present a map of where they're doing the various surveillance throughout the state, and if there is COVID coming up that is expected or unexpected. And this is from today, looking at Melbourne, our major city, where there's an example here of an area where there's a positive detect in the sample from yesterday, or from actually this one's from last week, which was expected. And what they mean by that is that they know that there's some cases living in that area. However, today, for the first time in probably a year in the town that I'm currently in, was the first positive unexpected detect in a small town of about 500 people. So the question is, there is someone excreting or contributing to the wastewater with a SARS-CoV-2. However, that person may not have been identified. And so that then means that there will be a whole lot of public health activity and testing in Apollo Bay to identify that one person who is not known to be currently a case of COVID. So this is happy to answer any questions, but really an introduction to why we thought it might be useful to look at the feasibility of wastewater and environmental surveillance in Indonesia, and the work is being done where you are in Yogyakarta to support the public health response during the pandemic, and how can we use this to help the public health officials decide where to target various activities around population or community surveillance, perhaps vaccination, and provide further updated information. So thank you very much for that, and I'll unshare my screen.
Thank you, Julie. That's perfect, and perfect timing as well. Thank you for that. So for those of you who are not familiar with the wastewater, so we do have a wastewater system in Indonesia, even though not all households would have it. But that's also the background why this is a potential way to do surveillance, which is probably cost-effective and low cost compared to individually testing and tracing everyone in the area. And also, we are looking for the next pandemic in a few years, maybe not so soon, hopefully, but COVID-19 would definitely not be the last one, right? So that's why these all knowledge are important, not just for COVID, but also potentially for future uses as well. Thank you, Julie, again. And I would appreciate if you would hang out and until our Q&A session at the end of today. Not at the end of today, at the end of this session. So I would like to welcome Dr. Indah Kartika, who's really, really smart and really, really nice. I think those combinations are important in the researcher and lecturer. So Dr. Indah Kartika is a pediatric cardiologist. She's also leading the wastewater surveillance in Yogyakarta in collaboration with MCRI. So she is leading what would be a groundbreaking one in Indonesia, and Yogyakarta is the groundbreaking one in Indonesia. Dr. Indah Kartika also finished her PhD from Melbourne University, and I had the privilege to study with her when I did my master's there as well. So we went a while back. And Dr. Indah, I would like to give you the Zoom to you, and you have 15 minutes for the presentation. Thank you, Doc.
Tiara, let me share my screen. Can you see it?
Yes, your sound is a bit small. Is it possible to turn it up a notch, Doc?
Well, thank you for coming, and thank you for the organizing committee for giving me opportunity to share in this important meeting. I would like to share our project on SARS-CoV-2 surveillance using wastewater and environmental sampling in Indonesia. Well, This is actually our team, both from MCRI, Prof. Julie Bynes, and also from the Monash University. And from Indonesia, we have Dr. Ida, Dr. Jarir, Professor Titi, Dr. Vika, and our other team members. Well, as have been emphasized by Professor Julie Bynes, that the wastewater surveillance of COVID-19 would have many benefits and also may be cost-effective in low and middle-income countries like Indonesia, when diagnostic testing capacity may be insufficient. And this project is actually aimed to inform the impact and the feasibility of a program of the wastewater and environmental surveillance in Indonesia. And we would like also to monitor the community burden of SARS-CoV-2 infection, and if it is possible, to be an early warning system for the early identification of SARS-CoV-2 outbreak within a community. And we did a routine surveillance samples of the wastewater and soil collected from the sewerage and also the wastewater and environment at 10 predetermined locations, including near source tracking, were taken every week and also fortnightly using the grab and passive sampler. And this sample then are transported to the microbiology lab in Gajah Mada University for the processing and analyzing for the SARS-CoV-2. And we analyzed the data by presenting the positivity of SARS-CoV-2 from the environmental sampling and also the trend over time sample positivity and COVID-19 cases as well as other indicator. And we will correlate between the viral load in the wastewater sample and the incidence of COVID-19. And we would like to also report this result to the local public health office and sewerage management facilities to allow the intervention. And in Yogyakarta, we actually have two systems of wastewater management, including a center wastewater management plan, or it is called by the Instalasi Pengolahan Air Limbah or IPAL Sewon, that is managed by the provincial government, and also the community wastewater management system or IPAL community that is independently managed by the local one. And the service coverage of the government wastewater treatment plan include 13 of the 14 sub-district in city of the Yogyakarta, and also 4 of sub-district from the Sleman and 3 from the Bantul. And we have discussed the site location of the samples at our centers and also with our colleague from the MCRI and Monash University. And there are two types of the samples, including water and soil, or in combination with water. Water samples are collected from the manholes, river, and public swimming pool, as well as the IPAL Sewon and near source tracking water. And from the near source tracking, we take the samples from the community spaces, including traditional market, boarding schools, flat or apartment or hotels and the malls. And part of the city, as you know, and the rural areas also manage the human effluent via the septic tanks with toilet or perhaps by open defecation with the possible contamination of the surface water and rivers. And therefore, to understand the level of contamination of the SARS-CoV-2 in environmental that may reflect the presence of the community infection, we also select this near source tracking sites. And we have decided to choose only 3 of the 5 districts in Yogyakarta province, considering the coverage area of the wastewater system and its populated areas, as well as the geographical purposes. And of these 10 sites, we select 6 sub-districts from the Yogyakarta city, considering the most coverage area of the provincial wastewater system and urban sewage system may provide more representative samples of the community. And we also select 2 sub-districts from sites of Bantul and also 2 from the Sleman. And we collect the samples every week in 3 sub-districts in the red box, and while others we collect fortnightly. And we did site simulation prior to sampling collection to see how is the feasibility to take the sample. And in this simulation, we learned of the wastewater sample process, as well as learning how to correctly take the sample using SOP and timely transport the sample to our lab. And we did also the training to the laboratory staff First was the internal lab training, and we share and learn the SOP for the sample concentration before proceeding to RNA extraction and the measurement. And during this internal lab meeting, we also collected the question, and also we get some discussion and also feedback from the Melbourne team. And regarding the dissemination process, we have done several meetings with the stakeholders, and we have conducted a meeting with the Public Health Authority to ensure the availability of the COVID-19 data from them in corresponding catchment area. And we also did a meeting with the official government and also the some faculty in the university and other stakeholders to make them familiar with our project and also provide the feedback about the sampling point to take and also the expected outcomes. And during the meeting, they suggested to take the samples also from the communal wastewater treatment plant due to the majority of the capacity of the government wastewater treatment plant coverage is limited to the city of Yogyakarta. And for the data entry, we use the RedCap database, and we have 6 arms available on our database consisting of the baseline characteristic of the sampling point, sampling collection detail, lab initial data, as well as the PCR result and the COVID-19 cases from the website and also the health office data. And our team also developed a video containing the introduction of the our project and also the team. And we also developed a handy module and also a handy field guidebook containing the field scheduling, also the number and location of the sampling point, and also field SOP. And finally, our first day of the sampling was on July to the 27th, and it is when the COVID-19 cases and the death rate were at the peak in Indonesia as well as in Yogyakarta. And until now, we have conducted wastewater surveillance of the SARS-CoV-2 in Yogyakarta for more than 10 weeks, accounting for around 560 samples. And every week we collect around 50 to 60 wastewater samples. And overall, just under 60% wastewater and environmental sampling were positive for SARS-CoV-2 RNA. And it can be seen here that the number of confirmed COVID-19 cases of incidence of COVID-19 is decreasing, as well as the death and the positivity of the engine SARS-CoV-2 RNA in the wastewater, and also the environmental sampling is also decreasing according to the incidence of the COVID-19 in population. And in the first week, as you know, that in the first week of the sampling collection, we found around 80% of the positivity for SARS-CoV-2 RNA, and it is steadily decreasing over the week. And we found that around 80% of the sewage using the grab sampling were positive for the engine, and from the NST water using the passive sampler, approximately around 60% were positive. And from the river sampling using the grab method, it was around 60% positive. And so far we found 2 positive engine for the SARS-CoV-2 from the soil sample. And it is quite similar with the ORF gene, but we found 5 samples of the soils were positive. And as we take the samples weekly as well as the fortnightly, we aim to present the data based on this basis as well. And for weekly basis of sampling collection, we are showing to you that the engine copies per mil for the sewage and also the river, and also the engine copies for the NST, as can be seen here, that it seems that this is similar, the trend of decreasing the engine's copy for the manhole using the grab sample with the confirmed COVID-19 cases. But it is perhaps quite different. If you can see here that it is the engine copy using the manhole and the river, the trend quite similar with the incidence of COVID-19 per 100,000 population. But it is quite different with the NST water, because the NST water using the passive sampler, it includes septic tank, including at the COVID-19 shelter for the isolation, also traditional market, and also mosque, and also factory and local government building. And for the sampling collection using this passive sampler, in the 4 weeks earlier, we found that the engine copies are increasing as compared to the confirmed cases. This might because we took the samples from the septic tank and there might be any accumulation of the RNA in this site, and also perhaps there is any different type of the sampling collection using the passive one. And the trend, it is quite similar also for the ORF gene. And when the number of traditional epidemiological indicators are decreasing, the ORF gene also showing to you decreasing. And NST water also using the passive sample tend to increase. And this is also the case for the result of the fortnightly data, both for the engine and also for the ORF. So we can see that the incidence of COVID-19 is decreasing, and the engine copies from the manhole, also from the NST water, also quite the same with the ORF. Well, to evaluate the effectiveness of the wastewater treatment and of the wastewater effluent, we also compare the engine copies per mil from the inlet or from the inflow, and also from the outlet or outflow of the EPAL-7. It can be seen here that the engine copies per mil from the inlet are positively high, and at the outlet of the EPAL-7, the engine copy is almost undetected. It may reflect that the treatment is quite effective enough to manage the wastewater coming to the EPAL-7 system. And actually, we are still discussing what is the best to present the data, including also we will use the map, and we use the cutoff to determine the transmission risk in the area using the CDC recommendation, and it is considered to be low when there is less than 10 new cases per 100,000 persons in the past 7 days, and moderate if when 10 to less than 50, and also substantial if 50 to 100 and more than 100, it is high. And this map is actually trying to show you a similar trend with another version. And we have collected a sample from the 10 sub-district sites across Yogyakarta. And the gradated blue shows the number of newly confirmed COVID-19 cases per 100,000 persons in the past 7 days. And the red and also the green one is actually showing the positivity of the SARS-CoV-2 RNA using wastewater and environmental sampling. And the red is meaning positive, and the green is meaning the negative. And as can be seen here, that from the engine in the first and the second week of the data collection, we found more red than the green. And until in the last week, we found here that in the last 8 weeks that we found more green than red. So which is showing more negative SARS-CoV-2 RNA found in our surveillance that it is also similar to decreased number of the confirmed cases in that week. Well, this map is actually reflecting similar message from the ORF gene positivity. Well, I think that's all. Before finishing my presentation, I would like to express my greatest gratitude to all these people who made this project happen. And thank you very much for your kind attention.
Thank you, Dr. Ida, for the presentation. For the audience, I know it's a lot to take in. So feel free to jot down a clarification question or anything at all that you would want to make clearer about both Julie Bynes and Dr. Indah presentation. It could be in English, could be in Indonesian. It could be as complicated as possible, and we will try to answer it, or it can be as simple as possible. That usually the simple questions are the hardest one, I find. So before we move on to Dr. Fika's presentation, I just would like to remind everyone again, please do type in your questions or prepare your questions for the Q&A session that will be coming up after Dr. Fika's presentation. So Dr. Fika, finished her medical training from UGM in 2007, and then she went on as a master of in epidemiology, and she got her MPH from Melbourne University, and then proceeded to PhD in research on vitamin D deficiency, mostly in children and pregnant women. But then she moved on and just joined the public health department as a lecturer. So congratulations for the new position, Dr. Fika. And now she is a co-principal investigator along with Dr. Indah Kartika on the wastewater surveillance for COVID-19 in collaboration with the Melbourne team as well. And Dr. Fika would now present one of the most important points in the surveillance is, so what next? What is the future direction of the wastewater surveillance, and what can we do about it, or how do we utilize all the knowledge and technology in our future pandemic? So Dr. Fika, I would like to welcome you, and please do present for 15 minutes, and the floor is all yours. Thank you, Dr.
Thank you, Dr. Tiara. May I share my screen?
Yes, please.
Okay, thanks everyone for having me today. And today I would like to present about the future implications of the wastewater and environmental surveillance for SARS-CoV-2. So as we may have known that in the early of 2020, around 11th of March, 2020, the WHO has declared that we are facing a global pandemic to COVID-19. And as you may see from the picture here, there are cases increasing by months during the 2020. And many countries around the world, they established new surveillance for COVID-19, but it is very challenging to set those surveillance because it is in fact, COVID-19 is very, very infectious disease with high rate of transmissions. And even for a symptomatic individual, they can transmit the disease to other people. And asymptomatic cases, especially for the asymptomatic virus carriers, they causes the clinical examinations and laboratory testing lag behind the disease transmission in the community. And also, especially in the resource limited setting, there are limited testing capacity and challenge around turnaround time when the testing was done and when the result is available. And that's why in many countries around the world, they start to combine the wastewater surveillance or the water-based epidemiology to serve as the complement for the routine surveillance. And we may all know that the impact of COVID-19 pandemic has been very catastrophic, and it has caused the global domino effects, which is causing the disruptions to the functioning health system. Many health systems are overwhelmed with the surge of the cases in the community, and also it also threatened the public health and safety and accessibility to healthcare. So many non-COVID cases being less prioritized during the surge of the cases. It also has implications to the food security as well as the stability of economy and financial institutions due to maybe, for example, lockdown. So those people who are earning their salary as a daily income, they may be disrupted because this COVID pandemic. And other challenges that we are currently facing are how to track the initiations, the spreads, and the changing trends of COVID-19 at populations-wide scale. And there is an urgent need to map the distribution as well as the magnitude of COVID-19 in real times. And we need to also apply large-scale applications for diagnostic testing, especially to provide fast survey in large populations to catch up with the COVID transmissions in the community. So this is where we place the water-based epidemiology or the wastewater surveillance, because it may help to targeting the populations at risk, because it's not only reported the symptomatic cases, but it also capture the information for asymptomatic cases. So it's less likely to be biased by the health-seeking behavior and the testing rate. Also, it can help to guide target public health for mass messagings and allocate resource, for example, testing in subpopulations or do the vaccinations, like targeting the vaccination group, and inform for infectious control policies such as limiting size of gathering during the, for a wedding ceremony or funerals, and also have to help the guide to building and building opening as well as the school opening. And it also helped to minimize the occurrence of surge of serious cases as early warning system, as has been mentioned by Dr. Ita previously, that may cause the overwhelming for the the health system, the healthcare worker, and also overwhelming the critical care hospital capacity. It also helped to prevent from the unnecessary long stay at home policy that can stress both humans and as well as the economy. So we can do more targeted lockdown and maybe in sub-district level rather than doing the large-scale lockdown, which is more stressing for many of people as well as the economy itself. And many studies has reported that it has provide cost-effective infection information, especially for large populations, because usually, for example, when we test one manhole, it represents a number of people or maybe household that is within the catchment of the manhole itself. And it's also important when the disease eradicated, where there's a low number of cases in the community, this kind of surveillance can help to identify any newly emerging cluster within the community. So these are the possible collection scale for the water-based epidemiology. Some study using the wastewater treatment plants, which is mentioned by Dr. Ita previously, the IPAL or Instalasi Pengolahan Limbah in Sewage, Bantul. And this will provide information of the infections that are happening within a population, maybe larger population that's served by the IPAL. And some study using the sub-sewer set. So it's just like a small version in the subpopulation. So they're like getting information for the pipes connecting between household and several buildings and try to identify the transmissions within that subpopulations. But it is a big challenge compared to the IPAL because we need a clear map about the pipes as well as the number of population that's served by the pipe or number of households that's served by the pipe. And some using at the facility level, for example, using COVID to track COVID infections in a person that work or as well as living in an individual facility, for example, in the hospital, school, dormitory, or university, and something like that. So these are the implementation of wastewater surveillance in other settings. I'll show you some from the high income country, for example, in Australia, and we may ask more from Prof. Julie later. So they actually combine the community surveillance, like using this information in their website. So they were telling people like who are being infected and where he or she has been around before the tested. So all people who have been who went to the location at a certain time, they need to go for further testing for it's a part of the contact tracing. And also they have combined it with information from the wastewater surveillance, like here, it is saying that the fragments of the virus detected in sewage in some suburbs, and they all blend all these informations and put informations in more informative to public, like using the public health order and saying that maybe the restrictions extended for several sub-districts in those locations. These are other implementations in the US, in Utah, and they actually put mapping here the information, and the round here, the round shape in different color, it's showing the trends of the sewage SARS-CoV concentrations. The reds indicated increasing, and the green showing a decreasing trend, and they try to align the information from SARS-CoV to the cases in the community and showing that it's pretty much alike, the trends between the sewage informations as well as the daily number of cases in the community. In the low and middle income country in Bangladesh, they actually apply similar system, but they are using a different style. Like they are using map here with different number, like the gradation of the map indicate the number of cases per week. And if you see the round here, it's pretty similar with the Utah one. So it gives information about the concentrations of viral loads in that locations, whether it is increased, low, or medium, or high. And they also align here the informations from the COVID cases and compare with the finding from the wastewater itself. And also put note here, so it's to avoid public panic. So they put that that's only tracked for the death virus in the sewage rather than live that virus that may can like causing infections. And this is the global data for wastewater sphere or WasteSphere. We are part of this collaborative working group. It's basically, it mapped all the wastewater surveillance around the world. As we may see here in United States, there are a number of locations have been joined this WSphere, and also in the Europe. And we have here in Australia and Indonesia here. It's actually, it is a part of our working group, and the data will be coming soon because we still try to combine and do some adjustment with our data, so it will be able to also be presented in this map. So this is basically, if you want to see, for example, in Wisconsin, and you can see what happened around the locations, whether the concentrations of SARS-CoV in several locations may be increased or decreased. And they also present the concentrations using this figure to showing whether any increase by times. Other applications implementation that have been mentioned before is using the facility-based wastewater surveillance. For example, here is for a college or university, and they provide information of the survey itself sharing by using email or text, as well as using their local dashboard. And they follow the result from the surveillance by doing the testing, for example, in the dormitory of the students, and more stringent health protocol, as well as allow for social distancing and reducing number gathering sizes, as well as moving to offline classes. And this kind of system is really rely on the ability of the college or university to make the test mandatory for students, as well as it's very rely on the student compliance for testing requirement and the willingness to do the self-isolations. So this is basically a framework. When we want to establish the campus wastewater monitoring system, we need to have the information about whether we want to identify the SARS-CoV trend over time and what are the follow-ups, such as doing target individual testing, contact tracing, or vaccinations. And also we need to identify the map sewer system within the campus and the accessibility to the sampling sites. And whether we want to do it like weekly, fortnightly, or maybe daily, it needs to be decided prior to the establishment of the system. And also the more important thing is the how to interpret the data and use and how we engage with the public health to contextualize the data. This is one of study in North Carolina. So it's doing 90 on-campus site wastewater-based surveillance, and they did thrice a week data collection, sample collections. And it is combined with in-house tracing, symptomatic tracing, and also daily symptom reporting. And they reported that from the data collections, the result will be available around 30 hours since the collections. And this system was able to detect a single asymptomatic individual in the dormitory of the campus within, and the dormitory itself have around 150 and to 200 people, and then they will allow the isolations further for preventing for the spread of the virus. So basically, they test the water. When they found positive result, they check whether is there any positive cases, some maybe student that recently have positive result. If it is yes, that means that shedding and the student itself has been doing isolations. And then if not, they look further whether any close contact in the location and whether the close contact has been doing the self-isolations. If it is no information, they try to find whether any students with recent major symptoms are related to COVID-19. If it is not information, that means they will do the search task building, because it may be somebody with who are asymptomatic, have been around and blend with people, so he or she may cause the infection spread. And other potential applications of the water-based survey itself, it is still being discussed in the some editorial of the journals, like I displayed here 2 journals. So they can use this kind of surveillance to monitor the national vaccine effort. For example, they can help to provide the early warning system to prioritize the hotspot when the vaccine coverage is mostly urgently required. And also it helps to understand the level of ongoing transmissions in the community that is monitored by this system, and provide insight on the impact of the vaccination program itself. And also provide evidence about the SARS-CoV RNA viral loads and also viral shedding trends during the rollout of the vaccinations in the community. So although we have seen a lot of advantages of this kind of system, there are a number of challenges that we are still facing. For example, Indonesia, like what we face currently in Indonesia, that majority household in the lower and middle income country are not connected with the sewage networking. So we need to sort of like modify and adjust with the the local situations and condition, like Dr. Ita previously presented. Maybe we also using the local IPAL or water treatments like community IPAL komunal. And also there are challenges about the interpretations, communication, and the use of the result to avoid over reporting or like under reporting of the result. And some suggest that maybe using positive and negative result during health season, and using the exact concentrations of viral RNA in low seasons. Some challenge, especially in developing country as well, is the supply chains delay for the kit, RNA kit, and also the the human resource itself, and the turnaround time. Sometimes it's a bit tricky because the process takes a while as well because it needs the filtration and so on. And it needs a close collaboration, so between the scientists who generating the wastewater data and the public health agency, as well as the wastewater utilities and expert in communications and data visualizations to prevent false assumptions or under and over interpretations before communicating all of these informations into public. So to sum up, water-based epidemiology could complement the current community surveillance for COVID-19 cases, and it helped to detect ongoing transmissions in the community and confined spaces such as school or dormitory. But it needs collaborations among stakeholders for data interpretations, and especially to communicate the result with publics. Thank you. Thank everyone to have contributed all great work for the projects, and thank you, Dr. Yara.
Thank you, Dr. Fika, for the really good presentation, and that you have portrayed how it has been implemented in both high income and low middle income countries. I think this is a really good visualization of what can be done and what are the challenges. And I'm also happy that some of the audience here have jotted down their questions in the chat box. So I'd like to spend the next 35 minutes on just focusing on these questions, and also if Julie Bynes, Dr. Indan, Fika would like to add on, some comments and things that you think would be beneficial. So the first question would be on the, I think there are two questions pertaining to the selection of the sampling site, and how did you went about selecting the sampling site, maybe both for Melbourne, and also what modifications were done in Indonesia, because Dr. Fika said all the challenges relating to the sewage system. Maybe Dr. Inda, you could go first, and then we go back to Julie.
Okay, thank you. So the question is that about the decision to select the sites. So we decide to select the sites include the coverage area of the center wastewater system at first, and we consider that why we take more 6 sub-districts in the urban area, because the urban sewage area is more representative for the urban sewage system. But we will take also the samples from the community wastewater treatment plant of the IPAL community, because some of them also was not covered by the IPAL Sewage. And also the consideration will be about its populated area as well, because we actually have not the exact number of the population that may representing the sampling site, but we do have the number of the coverage of IPAL wastewater coverage in its sampling site. So yeah, it is actually one of the limitations of this. But we also consider to take the sample why it is only in 3 districts of the 5 districts in Yogyakarta. Also, it is about the geographical purposes. Yeah, I think perhaps.
Does it have anything to do with the COVID cases in the area, Dr. Indah?
Yeah, I think the COVID incidents at that moment in March, while we try to select the sampling site, is actually it's quite similar in 5 districts of the Jakarta. So for I think it's quite similar for the incidence of the COVID-19, but we see in the urban area, because the risk of having transmission is quite high compared to the rural area. So we also concentrate more in this area of the sampling collection.
I see. Okay, thank you, Dr. Indah. Maybe Professor Bynes, would you like to add on to that on the sample selection? Thank you.
Yeah, so sample selection, I mean, the idea of this is to get as good a representation of what's happening in the community. And one of the challenges for the scenario in Jogja is not everyone is connected to the formal wastewater network. And so how can you obviously capture that information and try and move upstream as far as you can in that, to be able to identify subpopulations that may provide information and be able to target your public health network. But also then I think what the team have really tried to do is, for some of those other areas where you don't have a formal wastewater system, what sort of things might be helpful to add to that surveillance? Will a small number of places connected in that region provide an accurate measure of what's happening in the community? Or are there other things like sampling river or soil provide additional information of that population, knowing that that SARS-CoV-2 often has its impact on people that may not have a lot of resources and also may not come for testing. So you really want to be able to have this surveillance system being able to capture as much of that diversity as you possibly can. So the team have done a really good effort, and trying to also be flexible if something's not being helpful, how to change and adapt to that.
Yeah, and that's why we see the manhole, the river, the soil, and everything else, which is might be the case in high-income country and sites, including Melbourne, right? Cool. Yeah. Thank you so much.
Exactly.
Yeah. And we have a lot of field epidemiology training program students in the audience, so I'm sure, and they have jotted down some interesting questions as well. The next one would be for Julie. So COVID-19 infection from wastewater is still unknown. Would you then suggest the water treatment company in the region, to avoid the infection risk? But also maybe you have mentioned about the chlorine and
Yeah. So it's thought, there's far more evidence for respiratory transmission of SARS-CoV-2. So to put it in context, there has been potentially a small number of data on the potential for either aerosolization or transmission from people's feet or something like that for SARS-CoV-2 from contaminated environments. However, the magnitude is probably very, very small considering to aerosol and respiratory spread. If you're talking about formal sewage systems, and Dr. Indah presented very nicely, the treatment in the formal sewage system seems very effective in the Jogja area and also in the context of Melbourne where I live. So, things like the normal treatments in those treatment plants, as well as, let's say, domestic treatments using chlorine and bleach are very effective against SARS-CoV-2 in terms of surface elimination and so forth. So that's the way I think that the potential is more around close contact in households and where all of those issues may not be quite as stringent.
Yeah, thank you. Yeah, and this is where Dr. Indah showed that the inlet gene was high, but the outlet one was almost undetectable, meaning that along the way, the virus had died. Yeah, hopefully from the treatment.
Yeah, and which is what a treatment plant is meant to do. But we were a bit cheeky, and we really wanted to prove that that was the case by taking the outlet as well.
Yes,
so that should be reassuring.
I'm also happy to look at that. Oh, that's good. Okay, it's working. Right, and Dr. Indah, you've been working closely with the government of Yogyakarta and Dr. Fika as well. How that be? Were they okay with you, collecting feces and stuff? What was the government response?
Thank you very much, Dr. Tiara. So actually, we have done several meetings with the government as well as the health office, and we will also report these results to them. But the wastewater surveillance maybe can be effectively used, I think, as a perhaps preventive or perhaps in Melbourne, perhaps as a early warning tool, because as soon as the virus is detected, then this should be taken as a signal of the possible emergence or perhaps re-emergence of the pandemic. And the second one, perhaps about the management tool, because the absence of the virus in wastewater may indicate perhaps that is a corresponding catchment area can be considered as still low risk at the time of the and perhaps the third thing, perhaps consider FTP safety thing, because if the virus is detected in the wastewater but the testing of the community or population is negative, then the investigation of the undetected infection sources perhaps should be undertaken. This perhaps can be used for
I would like to add a bit information. Like currently we are doing the proof of concept. We're trying to prove our concept as also about feasibility to conduct this kind of surveillance in our settings. And we're still thinking about what is the best way to report the informations and try to play with the data. What is the best data visualizations to be shared with publics. And if may remember for the map that was presented by Dr. Ita before, we can see the gradations of the blue from dark blue to light blue, and also the positive and negative, the red dots and the green dots. And maybe we should be aware when it is like the number of cases is, like the map is light, and then suddenly there are a lot of red dots within the map. So that may be sort of like information that, it is something that might be coming in the near future. Maybe there's a number of increased cases in that part of area.
That's interesting, because we always talk about research translation into policy or programs, right? So this is research into translation into policy in action. So you have engaged the Jakarta health offices, and some of the participants are also from the health policy and management, and which they would like to learn more about how you engage the stakeholders and make them understand how important this is and whether this is cost effective and stuff. So thank you so much for that answer. And the next one would be, is it still effective to use water-based surveillance when COVID cases are still high? It says in Indonesia, the polio eradication approach used environmental surveillance because it's almost undetectable within the population. Would anyone would like to take up on the question? Thank you. Oh yeah, Julie, please.
I'm happy to take that up. Look, I think there are a couple of different ways you can interpret this. This project is all around feasibility of where a wastewater system will work. And the best time to start it was actually when you had a lot of cases, because we found a lot of positive detects, and we knew the system worked and we could actually collect and identify, and 80% of all was remarkable. how much positivity there was there, and that very closely matched what was happening in the community. I think in countries like in Australia, in Melbourne, we had very few cases. Now we've got lots, but it has been used as an early warning. Just like in my little town now, early warning, people go and get tested. And so that's in our scenario. But in your scenario, there is a level of comfort if you're seeing the cases coming down and you're seeing the detects coming down, because one of the problems is that are the case numbers truly reflecting what's happening outside? Are people still coming for testing? Is there still asymptomatic spread, or people with mild disease not having? And so it's another way of checking up without having a huge burden of individual tests going through. And also too, if you follow Dr. Inda's map, and then all of a sudden those green places become red, you'll get a feel for the data of what, what the balance might be. If you suddenly see in places previously green, red, then you might expect a week or so later those light blues will become dark blues. And so that's really, it doesn't necessarily need to be zero, but you need to understand where your baseline is and go from there. So it's still useful information, particularly as you're rolling out vaccination and targeting other public health messages. That's in my view anyway.
Cool. Really, that's really interesting. So this is a feasibility study, and then we would like to see how, how possible it is in Indonesia setting and Yogyakarta setting. That's very important. Maybe Dr. Inda and Dr. Fika, would you like to add on to that, or we're good? Okay, so considering that the COVID-19 pandemic would be shifting to endemic phase, and I'm really sorry about Melbourne, Julie. I know it's 1400 today, and Melbourne has been in a never-ending lockdown, which is, I think, the stress of the lockdown is a bit more than the actual, burden of disease per se. Well, Indonesia, we have, when we have 50,000, you're like, okay, we're fine. Anyway, so the next question is pertaining to it will shift to endemic phase. What's the feasibility and applicability of the wastewater surveillance, for routine surveillance there? And I think that's what we're trying to do. Maybe what aspects needed to be considered, maybe not just for Jogja, for other districts in Jogja as well. Maybe Dr. Indah and then Dr. Fika and Julie to add.
Do you want to answer?
So aspects, if you want to scale this up and then, make it as a routine surveillance program, what things do we need to consider so it's feasible, or maybe it's not feasible, and that's exactly what we're trying to prove now.
Yes, I think the engagement to the health office is very important in this kind of situation, because we will take the report to them, as well as perhaps we will do the sustainable surveillance of this wastewater surveillance of the SARS-CoV-2. If it is possible, then we think that it is quite feasible until now, because we do have resources, the manpower, as well as we can do this. But perhaps in the long term, we may not take all the weekly, or perhaps the fortnightly, or perhaps we will take the monthly, or perhaps the fortnightly for the wastewater sampling collection. And also because it is quite perhaps cost effective if we do the wastewater surveillance system compared to the traditional epidemiological indicators for the COVID-19. As you know that Professor Julie Bynes has mentioning that not all the patient, actually the asymptomatic one will do the testing. So yeah.
Right.
To scale up, also we need to engage them to have other cities perhaps will do this in the long term, I think. Yeah.
Yeah, or to sell it to the MOH, to the infectious disease directorate, and make it a yeah, either a bottom-up approach or a top-down approach, whichever works in Indonesia, I think. And maybe Dr. Fika, you would want to add on to what's the most difficult thing in the data collection for the water-based surveillance?
I think, yes, as we may know, that not all is connected to the system, the main wastewater system. So I think at the beginning, we have to identify the type of the wastewater system itself. Maybe, for example, we use the large one, and then we use the communal wastewater system, which is more in a smaller scale, between maybe around less than 100 to hundreds of people living in the community. And also in some places in Indonesia, we found that, for example, like in apartments, they usually have like their own system. So we sort of like can combine the use of water-based surveillance using the wastewater treatment plant, as well as into some facilities where confined people living in the buildings, to detect the fluctuations of the SARS-CoV virus in the wastewater itself. So that's a lot of things that can be done. And when the case is very low, I think it's good, because when it suddenly increased, then it will alarm us that something is going on and something will happen by looking at the concentrations of the gene copies of the virus itself. So I think it is important to keep going with the system, the surveillance, although the case has been maybe reduced.
Yes. So if it's cheap and doable, it's an ideal thing to do, actually, fortnightly or every monthly. And yeah. And Julie, do we want to add on to that? What do you see maybe in other countries and also in Melbourne?
Yeah, so I think that the important thing is that this is not, this is a development or assessment of a feasibility of a surveillance system that could be used for other things in addition to SARS-CoV-2. And so in, someone's mentioned polio. In a number of places, polio surveillance is done through wastewater, and the idea of picking up the odd case. But also too in Melbourne, they have a program that the public really know about this greatly, but around looking at antimicrobial resistance genes, around drug use, around a variety of different endpoints, looking at other things. And you mentioned at the very beginning about what's the next pandemic. If you have a system up and running, what are the, we'd be much more nimble to be able to look for new things if they arise, knowing that the system's in place and can be, pivoted to look at a different organism or a different factor. So I think COVID has really raised the profile of wastewater surveillance and the opportunity for wastewater surveillance for a number of applications, and it's really trying to understand what's relevant for your community to understand and how you can use that wastewater sample, in Jogja. We're looking at, thinking about typhoid. What can we find out about the prevalence of typhoid within the community using wastewater? Or is a variety of different things that you might be able to monitor in terms of things that are enteric in particular, or go through the gut and end up in the waste, that you might choose this type of system to monitor.
Yeah. So we do need to have a rigorous information on how much it costs and how feasible and what type of context it would work, really, right? And in terms of cost, do we have a calculation on that? Is it, or from the Bangladesh one, and whether it was economically sustainable for LMICs? I know in Indonesia, we are just trying to go about and assess that. If Dr. Fika or Dr. Indah might have information from other LMIC settings and whether this is something that could be sustainable.
I haven't looked at the more detail about the cost effectiveness. The many study reported that it's quite cost effective. For example, if you want to test rather than do mass testing for the populations, like a sub-district, for example, we can just select several manholes and doing the same test as the human test, but just we have like preparations like doing the water filtration before it can be, like the PCR can be done. So it is a bit more or less similar to the price of the one manhole price, similar to the price like a one person test. So it's from the perspective, our perspective. But well, we may do more, along with the study, we can do the, the cost effectiveness study later on, and to later decide and prove that the cost effectiveness of this system in our setting, especially.
Yeah, it is exciting to see what kind of public health responses that can come out of the surveillance, right? Whether when it peaks to a certain level, and then you're gonna have a localized lockdown instead of a whole Yogyakarta lockdown. And then you can actually determine, okay, we're free from the gene, and then we can have restriction ease up. And then rather than having everyone tested and, traced, which is, we don't have the resources as we have proven over the last year or 2 years almost. Right. Julie, do you want to add on to that? Just your thoughts.
No, I think it's important. We're very interested to see the sustainability, and that's based on cost. But then if you take it from the human cost or the economic cost of lockdowns are enormous. And if you would be feeling much more comfortable if you had no detects in a region to leave that region free to do its own business, right, and kids to go to school and all of that sort of thing. And so those are the sort of costs that would be nice to see in a cost-effective analysis.
Yeah, it's interesting when you go about and stray, and like 70% of Indonesian population are in the informal sector, so we really cannot afford strict lockdown. But also we cannot really afford having, our hospitals swamped up with patients either, right? In what we've seen during the Delta variant second wave. Okay, so there's also Dr. Suni here. Maybe a quick question on favorable wastewater treatment to prevent fecal-oral aerosol transmission to prevent COVID-19. But I think we kind of have established that within the wastewater treatment in Indonesia, it's quite effective in reducing the number of active viruses. So PDAM is doing a great job, and our IPAL is also doing a great job, ladies and gentlemen, to my surprise. Maybe I'm just a bit cheeky of that. All right. Yeah, I don't think we have any other questions. So I'm just going to open the floor if anyone would want to open their mic or jot down further questions. While we have Professor Bynes, Dr. Inda, and Dr. Fika in the panel. You can ask anything. This is right. And also, maybe how long would you are you going to do the trial in Yogyakarta for this phase?
We will take the sampling collection for 6 months. Yeah.
Okay, and the end line would be to prove the rates of positivity and all. Is that so?
Yeah.
Okay. Okay, and maybe last question, if no one else in the audience would like to ask. How do you proceed after 6 months in terms of engagement with the local government, and also in terms of more epidemiological studies that you would foresee if necessary to establish a community-based surveillance such as this one?
Well, correct me if I'm wrong, but it has been emphasized by Professor Julie Bynes that it is not currently possible to directly convert the concentration of the viral RNA that we take from the wastewater to disease prevalence in the community. But at least the wastewater surveillance of the SARS-CoV-2 may complement the data from these traditional epidemiological indicators, because sometimes they will have any biases, as you know, that perhaps not all people will get tested, and also will report that they may have any infection or symptom. Something like that. Perhaps Dr. Vika or Prof. Julie will add some more.
What can we expect from the team in the next after 6 months, probably?
Okay, so we think we expect that if it is possible, then we will continue this surveillance. Yeah, to be honest, because we are quite interested with this result, and I think it is perhaps very important to do. And also perhaps we will consider to also, Dr. Vika may add something, but we may consider to take the near source tracking as well in the school, perhaps, because as we know that now the lockdown, I mean some semi-lockdown is also open in Yogyakarta, so we may also see whether any impact on this school-age children, perhaps, or also the university. What about the surveillance of the SARS-CoV-2 in the wastewater in this area? I think that is will be very important.
That's one of the our current idea is just maybe to set the system in the school or university when they start the offline class, so it will give some informations if then there are a number of like positive or symptomatic cases go around the schools or maybe university, and may give some informations to move into maybe blended or maybe the online teachings method, something like that, maybe. Another thing that we as well, like I mentioned before, like to identify the target spot for increasing the vaccine coverage, when we found that a lot of COVID cases happening, a lot of concentrations of the SARS-CoV in the wastewater, that maybe we can identify some certain subpopulations and make a priority to prioritize the vaccinations in that regions. That can be useful as another applications of this system.
Yeah. Oh, yeah. Awesome. And that's a good outline for both policy and public health responses that can come out of the surveillance system. And of course, Professor Bynes have also mentioned that this can be applicable to a lot of things. And when we have a system, we might be a bit more resilient, to face the next pandemic in Indonesia and globally as well. So it's promising, and I really look forward to see, the progress of the team in Jogja, and hope to be able to more reports and how the government would take up on this, and probably you would like to expand to other provinces as well. And Dr. Indah Kartika could be the consultant for the Ministry of Health and the WHO as well on this regard. And I think it's important because a lot of things started from Jogja, and it's important to, kind of infect the rest of the nation with good, good knowledge and good application of knowledge. I think it's good to end when it's, in Indonesia we say sebelum dingin tungkunya. So when the stove is still warm, then that's when you kind of shut things off, and then people are happy, and then we're happy with it. And again, thank you so much for your time, for the three of you, Professor Julie Bynes, for making out time. And then I hope Melbourne would see a decrease in number of cases. I know that you will have 80% of vaccination in a couple of days, and hopefully that would subside, the case number down, or at least you can go about and not stuck, in your suburb. Dr. Indah, thank you again for the presentation. It's lovely to meet you again, and hopefully to meet you face to face. And also Dr. Vika, thank you so much for the insight and a lot of the knowledge from other countries as well. And again, we look forward to hearing about the team, and hopefully this is something that will go national and in our region as well. And for the audience who have been joining us, thank you again. I'm gonna give back the mic to the MC. Thank you.
Baik, terima kasih banyak Dr. Tiara dan Bapak Ibu yang kami hormati untuk memberikan apresiasi kepada pembicara kita kali ini bisa dengan memberikan reaction, klik reaction, tepuk tangan walaupun nggak ada suaranya. Tapi seenggaknya, tapi setidaknya Bapak Ibu pembicara dan juga moderator bisa melihat ucapan terima kasih kita pada kesempatan kali ini. Dan untuk kenang-kenangan, mari kita lakukan foto sesi bersama-sama. So, Professor Pines, we are going to have online photo sessions. If you don't mind, you might join us as well. Dan kami mengundang Bapak Ibu peserta untuk menyalakan kameranya juga. Kami tunggu. Sesuai aba-aba dari saya, kita mulai Bapak Ibu. Professor Pines, on my count. Ladies and gentlemen, in 3, 2, 1, cheers. One more time. Sekali lagi Bapak Ibu, in 3, 2, 1, cheers. Dan yang terakhir bisa kita beri gaya bebas, walaupun tidak sebebas biasanya. In 3, 2, 1, cheers. Terima kasih banyak. Sekali lagi kami ucapkan kepada Bapak Ibu peserta yang sudah hadir pada kesempatan kali ini. Nantikan seminar Rabuan setiap minggunya. Informasi terupdate bisa didapatkan di berbagai sosial media Prodi Magister Ilmu Kesehatan Masyarakat Universitas Gadjah Mada. Atas nama panitia yang bertugas, saya memohon maaf apabila terdapat hal-hal yang kurang berkenan. Terima kasih, saya Karisma Dewi pamit undur diri. Wassalamualaikum warahmatullahi wabarakatuh dan sampai jumpa.
Thank you again, Julie, Dr. Indah, Bapak Tika, Bapak Ibu. Halo Pak Wahab.
Terima kasih. Terima kasih banyak semuanya.
Halo Mbak. Telat jawabnya. Mbak Indah yang dari